Hello everyone, my name is Felix.
And I'm Hannah.
We're going to teach you guys how to make your own antibiotic-resistant bacteria today. So before we get into the specific steps as to how you can create your own resistant bacteria, we're going to go over the two major classes of antibiotics that you can make your bacteria resistant to. So the first major type are beta-lactams. These guys operate by targeting and attacking the peptidoglycan layer in bacteria cells. So peptidoglycan is this protein carbohydrate mesh that lies around the periphery of bacteria cells. They help keep bacteria structurally stable and resist osmotic pressure from within the bacteria cells, thus preventing bacteria from lysing or exploding. So what beta-lactams do is that they target and disrupt this peptidoglycan layer in bacteria cells, thus causing bacteria to lyse and explode and die off. Now, aminoglycosides, on the other hand, target and bind to the 30S ribosomal subunit that's found in bacteria cells, thus preventing them from making their own proteins. It inhibits protein translation, and without the ability to make their own proteins, bacteria have no choice but to die off. Now, you don't have to worry about aminoglycosides targeting the cells in your body, because human cells use a slightly different modified version of ribosomes that is different from what's found in bacteria, thus making them unaffected by aminoglycoside antibiotics. So, poll-everywhere question for you guys. Which do you think, gram-negative or gram-positive bacteria, are more resistant to beta-lactam antibiotics?
So think about this for a moment. I switch over to poll-everywhere. All right, I'll give you a few more seconds to answer. Okay, there's a 70-30 split.
Okay, so most of you guys chose gram-negative bacteria, and that is in fact the right answer. So the reason why gram-negative bacteria are slightly more resistant to beta-lactams than gram-positive is two reasons. So the first reason is that the peptidoglycan layer in gram-negative bacteria is a lot thinner than what's found in gram-positive bacteria. So this makes peptidoglycan a much less vital structure in gram-negative bacteria, thus making it so that beta-lactams targeting peptidoglycan in gram-negative has a much milder impact on the bacteria than in gram-positive bacteria. Now, the second reason is that gram-negative bacteria have an outer membrane surrounding the peptidoglycan. So this outer membrane helps shield the peptidoglycan from foreign molecules in the environment like antibiotics. So it helps provide an extra layer of resistance in that regard. Now, in terms of gram-positive bacteria, their peptidoglycan layer is really thick. It's also directly exposed to the environment where antibiotics exist in the environment. So it's pretty much open season on gram-positive bacteria if you introduce beta-lactam antibiotics into their environment. So we just went over gram-negative, gram-positive, their relative resistance to beta-lactam antibiotics. Now in terms of aminoglycosides, both gram-negative and gram-positive bacteria are directly susceptible to aminoglycoside antibiotics because they both have that 30S ribosomal subunit. So 30S ribosomal subunit, that's found universally in bacteria. So that makes both gram-negative and gram-positive, or pretty much all bacteria in nature, directly susceptible to aminoglycosides.
Now I'll talk about the principles involved in making your antibiotic-resistant bacteria. So we use induced mutations in our experiment. And spontaneous mutations are mutations that occur naturally in organisms that contribute to the genetic diversity. On the other hand, induced mutations happen when people or scientists interfere in order to lead evolution into a desired direction. So in this case, we apply antibiotics in the growth of bacteria in order to make antibiotic-resistant bacteria. So natural selection is the theory that helps induce mutations. Charles Darwin suggested the theory of natural selection, saying that the survival of the fittest. So without you having to try anything, the antibiotic-resistant bacteria will naturally survive, and the remaining population will eventually turn into antibiotic-resistant bacteria, so you won't have to do anything in the process. So now I'll talk about the exact procedures you should go through in order to make your antibiotic-resistant bacteria. So you should always keep in mind sterile techniques, since you won't be able to reach desired results without eliminating unwanted, undesired bacteria from your environmental setting. So before starting your experiment, you should clean your lab table or environment with alcohol. Then you should prepare these following elements. So you need Serratia marcescens or any kind of bacteria, antibiotics, discs, agar plates, pipette tips, a plate of ethanol, a loop, pincets, and a Bunsen burner. So you should start the experiment by culturing the bacteria onto the agar plates. So using the pipette and pipette tips, dispose constant amount of bacteria onto each agar plate. And you should use the spread plate method in order to diffuse the bacteria onto the disc after disposing them. So using the loop, you should try to spread it evenly onto the agar plate. And in the method of doing so, you should keep in mind sterile techniques. So in between, using the loop, you should always sterilize it using the Bunsen burner and the plate of ethanol. The disc diffusion method is not necessarily needed in this experiment, but it is a method used to determine the strength of the antibiotic, as we do not need to compare or measure the strengths of antibiotics. It's not directly related to this experiment. So in order to do the disc diffusion method, you should place soaked discs with antibiotics onto the middle of each bacterial cultured plate. So you should make sure to sterilize the pincets that you use in between every process. If you want to compare the resistivity and strengths of two different antibiotics, you should create a different set of agar plates and soak the discs on the other antibiotic and also place it in the middle. And you should measure the zone of inhibition in order to check the strengths. So the zone of inhibition is the circle near the center of the plate where bacteria can't grow because of antibiotics. So the larger the area is, the stronger the antibiotic has acted on the bacteria. So there are two ways of measuring the zone. You can measure the diameter of the zone or the distance from the edge to the starting point of the zone. So after waiting 2 or 3 days for the bacteria to grow, you can measure that in order to determine the strength of the antibiotics.
So the disc diffusion assay is pretty good for if you want to measure the amount of resistance in a bacteria population. But it's not good for growing your own bacteria because you're essentially clearing out a dead zone of bacteria as opposed to growing colonies. So here we're trying to grow our own antibiotic-resistant bacteria. So instead, we're going to use the colony count method. So in this method, we use petri dishes infused with antibiotics. So we're going to use the same exact methods as we did in the previous few slides. We're still going to plate bacteria onto a plate. However, the plate we're going to be using has antibiotic in it. And so only the antibiotic-resistant bacteria are going to be able to grow on the plates. So the little colonies you see over there, those are all antibiotic-resistant bacteria. So after growing them on the antibiotic-infused petri dishes, we can harvest those antibiotic-resistant bacteria and achieve our goals. Now, the colony count method is very versatile in that you can not only grow your own antibiotic-resistant bacteria, but you can also achieve the same goals as you did with the disk diffusion assay, in that you can also measure the relative resistance of a given bacteria population. So in that case, you take a sample from the bacteria population, you grow it on the antibiotic-infused petri dish, and the number of colonies that grow is directly proportional to how resistant the original bacteria population was to the antibiotic. And you do that by physically counting one by one the number of colonies on the petri dish. And so, in summary, there are 3 different breeds of antibiotic-resistant bacteria that you can cultivate. So the first one is beta-lactam resistant, so that's if we use a beta-lactam infused petri dish. And the second one is aminoglycoside resistant. That's if we use an aminoglycoside infused petri dish to grow our bacteria on. And then the third, in the master race, is the bacteria that's resistant to both beta-lactams and aminoglycosides. So that's if we use a mix of both in the petri dish that we use to grow bacteria on. So the last poll everywhere question: if you guys were to have a pet, would you rather have a dog, cat, or antibiotic-resistant bacteria?
You cannot go wrong with this question. Very nice large proportion of people want to have bacteria. Excellent. Do you have any more slides? Fine.
The right answer is antibiotic-resistant bacteria.
Of course, everybody receives credit for this question.
Thanks.
Thank you.